Welcome to part 3 of specific immunity. We're going to be taking a good look at humoral immunity right now. You can see that I brought forward the events leading to the activation of helper T cells. And I want to just remind you, as we're leading into humoral immunity, that the helper T cells, once activated, go out looking for, again, specifically looking for APCs that are displaying their specific antigen. Again, specific immunity. We're looking for specific antigen parts being displayed on these APCs. And that was just a reminder. Over here you can see that I have drawn some what would be B lymphocytes. And you can see that B lymphocytes, their cell membranes are just absolutely covered with receptor sites, and all of the receptor sites on each B cell are specific to one antigen. So you can see that the receptor sites between these three B lymphocytes that I drew, the receptor sites are a little bit different. These are kind of a U shape, these are a V shape, and these are kind of a rectangular shape. And so that's just to depict the very different specific antigens that each B lymphocyte then is going to be receptive to. Now, as it turns out, these receptor sites, again, are going to actually bind that antigen on the cell membrane. So let's just go ahead and do that. Let's just say that this particular B lymphocyte binds this particular antigen, okay? And yes, oh, they fit so nicely. They fit so nicely right down in here, okay? And then what's going to happen here at this B lymphocyte is that this B lymphocyte, even though we don't think of them as phagocytes, they will internalize this antigen. They will internalize this antigen, break it down into its little pieces, and then they will build an MHC2 complex so that they can then display a piece of that antigen on the cell surface in that MHC2 complex. Again, because it's part of the lymphatic system, a lymphatic cell, it would be an MHC2 complex that's displaying the piece of the antigen on the B lymphocyte. All right, well, so now we have what looks to me like an APC, and sure enough, it is. And so these activated helper Ts, we're coming back over here again, these activated helper Ts, again, are out looking for APCs displaying the specific antigen that they're able to identify. And so here comes this helper T over here, and sure enough, it's a match. It's a match. And so what happens at that point, since it is a match, the helper T binds to the APC, it's a match, and that helper T is going to start secreting, interleukin all over the place. Well, this interleukin then, the interleukin is going to cause this B cell to undergo clonal selection. Oh yeah, here we go. Okay, so again, they're going to clone themselves many times over, and don't forget, we'll also form a number of B memory cells over here. Okay, so by the way, this is the nucleus that I'm putting here in the B cells. All right, so clone themselves. Again, each of these B lymphocytes is only going to be able to detect or work against the specific antigen that we started with in this B lymphocyte. Okay, very specific. We are in specific immunity. Okay, So what happens next is that each of these B lymphocytes will differentiate into what we call a plasma cell. All right, so there's your plasma cells. Plasma cell. So the plasma cells then, each of them are going to produce absolutely millions of antibodies. And again, these antibodies are receptor sites and are going to be specific for the same antigen that we started with up here, that we were specific to. Now, these antigens will be released into the fluid, into blood, and hence our other fluids. And that's why we call it humoral immunity, because they're in the fluids. And as we discussed, the role of these antibodies is numerous, and we discussed that in class. So I'm going to stop here for humoral immunity, and I think I will make a part 4 just to pull it all together.